Identical twins begin their journey long before the mother who carries them has any idea they exist, long before any test confirms their presence, long before any heartbeat can be detected or any scan can reveal their growing forms. The journey starts with the egg. Every woman is born carrying all the eggs she will ever have, formed inside her own mother's womb before she herself was born. These eggs have waited, sometimes for decades, for their moment. Once every month, the female body selects one egg from the ovaries and releases it in a process called ovulation. The egg travels into the fallopian tube, a narrow passage connecting the ovary to the uterus, where it will wait for a maximum of 12 to 24 hours. If no fertilization occurs within that window, the egg is lost. But when conditions are right, when the timing is precise, something extraordinary begins. At the moment of intimacy between a man and a woman, approximately 250 million sperm cells begin one of nature's most remarkable journeys. 250 million, each one carrying within its microscopic head 23 chromosomes, half of a complete human genetic blueprint, each one moving with urgent, purposeful energy through an environment that is, at their scale, as vast and hostile as any ocean on Earth. The temperature shifts, the chemical environment challenges them constantly. Physical barriers filter out all but the strongest and most determined. The vast majority will never come close to their destination. Millions are lost in the first minutes. More are lost at each anatomical barrier they encounter. Of the original 250 million, only a few hundred will ever reach the egg, and of those few hundred, only one will ever enter it. That one cell, selected by forces science is still working to fully understand, presses against the egg's outer protective layer called the zona pellucida. Its head releases a precise package of enzymes that begin dissolving the zona pellucida at the contact point. The zona pellucida yields, the sperm head pushes through, and in the precise instant that the sperm head breaches the inner surface of the zona pellucida, something extraordinary happens simultaneously. A wave of chemical change, called the cortical reaction, fires outward from the entry point across the entire surface of the egg at remarkable speed. The zona pellucida hardens almost instantaneously, its protein structure cross-linking and changing in a wave that travels from the entry point to the furthest edges of the egg surface in a fraction of a second. Every other sperm cell pressed against the surface finds the barrier suddenly impenetrable. Their enzyme activity ceases, their flagella slow. They drift away. The egg has chosen its partner and has immediately closed itself against all others. Inside the egg, the sperm head releases its payload, 23 chromosomes drifting through the warm interior fluid. From the egg's own nucleus, 23 chromosomes rise to meet them. The two sets find each other with a precision that goes beyond anything human engineering has ever achieved, each chromosome identifying and pairing with its exact partner among the 23 incoming strands. Together, they form 46 chromosomes, a complete and entirely unique human genetic code. A code that contains every instruction needed to build a complete human being. Every physical characteristic, every biological tendency, every cellular process that will sustain a human life from this first moment to its last breath decades from now. This newly formed cell, now called a zygote, is the beginning of a human life. But for our story, it is the beginning of two. The zygote begins dividing. This is entirely normal. Every human pregnancy begins with cell division as the fertilized egg multiplies from 1 cell to 2, from 2 to 4, from 4 to 8, growing steadily as it travels down the fallopian tube toward the uterus. In most pregnancies, this cluster of dividing cells remains unified, one embryo developing from one fertilized egg. But occasionally, in a process that has fascinated scientists for generations and that is still not completely understood, the dividing embryo splits completely, cleanly, into two separate and equal cell clusters. Each cluster carries the identical genetic blueprint. Each cluster is complete. Each cluster is whole. Each cluster has everything it needs to develop into a fully formed human being. This is the moment identical twins come into existence, not in a delivery room, not with a cry, not with anyone watching, but here, in the silent warmth of the fallopian tube, in a moment of cellular division that takes place days before the mother has missed a single menstrual period. The split can occur at different points in the early development process. The earlier it occurs, the more separate the twins' development will be. When the split happens within the first 3 days after fertilization, each twin will develop their own placenta and their own amniotic sac entirely. When it occurs between days 4 and 8, which is most common, the twins will share a placenta, but each develop their own amniotic sac. When the split occurs later, between days 8 and 13, the twins will share both a placenta and an amniotic sac, living in the most intimate physical proximity possible throughout the entire pregnancy. Both cell clusters now travel the remaining distance to the uterus, side by side through the fallopian tube, two journeys that began as one, moving toward the same destination. The uterus has been preparing for this moment. Under the influence of hormones produced after ovulation, the uterine lining, called the endometrium, has thickened and enriched itself with blood vessels and nutrients, creating an environment precisely designed to receive and sustain new life. The cell clusters, now called blastocysts after a few days of continued division, arrive in the uterus approximately 5 to 7 days after fertilization. Each blastocyst is now a hollow sphere of cells, an outer layer of trophoblast cells that will become the placenta and supporting structures, and an inner cell mass, a small, dense cluster of cells at one pole of the sphere that will become the baby itself. Both blastocysts press gently against the uterine wall and begin the process of implantation, the trophoblast cells extending microscopic projections into the endometrial tissue, anchoring the blastocyst to the uterine wall, beginning the extraordinary biological communication between the developing embryo and the mother's body. The embryo releases specific hormones that signal its presence, preventing the menstrual cycle from continuing, triggering changes in the mother's entire physiology that will sustain and protect the pregnancy for the months ahead. The uterine wall responds, blood vessel growth intensifying around both implantation sites, nutrients flooding toward both new arrivals, the mother's body committing completely to two new lives it is only just beginning to know they are there. Both implantations succeed, both embryos are held, both are home, and now the most breathtaking construction project in all nature begins. At 3 weeks after fertilization, the neural tube forms in both embryos, the very first architecture of the brain and spinal cord rising from a flat disc of cells, its edges curling upward and fusing at the midline in a wave of cellular organization that establishes the fundamental structure of the entire nervous system. This fusion must happen perfectly. The genetic instructions that coordinate it are among the most ancient in the human genome, conserved across hundreds of millions of years of evolution. At 4 weeks, the heart beats for the first time. A cluster of cardiac progenitor cells in the chest region of each embryo begin contracting spontaneously, not because anything external has triggered it, not because any nerve signal has commanded it, but because the genetic program running within those cells has reached the precise moment when contraction begins. 2 hearts, each the size of a poppy seed, beat for the first time, simultaneously, in 2 embryos that 5 days ago were one. The blood vessels grow outward from the heart in branching patterns, reaching every developing tissue, establishing the circulatory network that will sustain this life. The placenta, growing from the trophoblast cells of the blastocyst outer layer, establishes its connection to the mother's blood supply, the umbilical cord forming as the physical link between the developing baby and its life support system. At 5 and 6 weeks, the limb buds appear, small rounded protrusions from the body wall that grow as dark pigmented circles beneath the surface, the nasal pits forming from the facial tissue, the upper lip completing, the jaw and chin taking shape. The brain grows with astonishing speed, expanding and folding, its different regions differentiating into the distinct structures that will eventually support consciousness, memory, emotion, language, and every cognitive function that makes us human. At 8 weeks, both twins are unmistakably human in miniature. Fingers have separated from the hand plates, the interdigital tissue between them having undergone programmed cell death to reveal 5 distinct digits on each hand. The face carries individual features. The major organ systems are established, and both twins are moving, spontaneous muscle activity beginning as the nervous system makes its first functional connections. They have been aware of each other since before they could be called aware of anything. From as early as 14 weeks, researchers have documented twins in the womb directing deliberate movements toward each other. Not random thrashing, not accidental contact, but oriented reaching. One twin extending a hand toward the position of the other. One twin orienting its face toward the sound of the other's heartbeat, transmitted through the shared fluid environment. By 20 weeks, both twins are fully formed human beings in miniature, floating in the warm golden amniotic fluid, connected to the shared placenta by their individual umbilical cords, completely enclosed in the world they have shared since the moment one cell became 2. They sleep together, they wake together. Their movements influence each other through the fluid environment they share. They touch, their hands finding each other in the warm fluid, their faces oriented toward each other, their bodies responding to each other's presence with the familiarity of the only relationship either of them has ever known. The months that follow are months of growth and refinement. The organ systems established in the first weeks now develop their full complexity and functionality. The lungs, which will not draw their first breath of air until birth, develop their intricate internal architecture, practicing their function by breathing the amniotic fluid in and out in rhythmic movements that exercise the respiratory muscles and prepare the alveoli for the moment they will need to process air. The brain's neural connections multiply at a rate of hundreds of thousands per minute, the architecture of a mind being built connection by connection. The senses come online. The twins can hear, can taste the amniotic fluid, can feel pressure and temperature changes, can perceive light as a diffuse warmth through the uterine wall when the mother is in bright sunshine. The eyes, sealed beneath fused eyelids throughout the pregnancy, practice their movements, rapid eye movement activity visible from very early in the 3rd trimester, the visual cortex of the brain developing its capacity to process images it has never yet received. The skin develops its layers, the vernix caseosa, a white protective coating, forming across its surface to protect the developing skin from the constant immersion in amniotic fluid. Fine lanugo hair covers the body, a temporary coat that will be shed before or shortly after birth. Subcutaneous fat deposits build beneath the skin, filling out the cheeks, rounding the limbs, giving the baby the characteristic appearance of a healthy newborn. By 35 weeks, both twins are fully developed, each one a complete human being ready for the world outside. The uterus, an organ of extraordinary elasticity and strength, has expanded enormously to accommodate both of them, its muscle walls stretched to their maximum, but maintaining their integrity and their function. The space between the two babies is now minimal, the contours of one visible against the body of the other, 2 lives pressed close in the warmth of the space they have shared since the beginning. The mother's body begins its final preparation. Hormones shift in precise orchestration, cortisol, oxytocin, prostaglandins, each one playing its specific role in initiating and sustaining labor. The cervix softens and effaces, its dense fibrous tissue loosening and thinning under hormonal influence, preparing to dilate from its closed position to the 10 centimeters required for birth. The uterine muscle, the myometrium, transitions from the irregular practice contractions of the third trimester to the powerful coordinated contractions of active labor, each one applying directional pressure that moves the baby downward through the birth canal. The first twin descends, head down in the most common and safest birth position, the skull entering the birth canal with each contraction driving it further through the passage. The skull bones, not yet fused at this stage of development, overlap slightly at the fontanelle spaces between them, the head molding its shape to navigate the birth canal. The passage is tight, the pressure is immense, but the baby's body is designed for exactly this. The compression that seems impossible is the precise pressure required. And then, light. For the first time in the entire existence of this human being, light. Real light, the overwhelming brilliance of the world outside the womb flooding the first moments of extra-uterine life. The lungs, which have been rehearsing this moment for months, take their first breath of air. The diaphragm contracts downward, the chest wall expands outward, and air rushes into the alveoli for the first time in history. The hundreds of millions of tiny air sacs inflate, a cascade of opening that transforms the lung from a fluid-filled practice organ to a functioning respiratory system in seconds. The blood flow through the heart shifts. The foramen ovale and ductus arteriosus, openings in the heart that allowed blood to bypass the non-functioning lungs during fetal life, close as the lungs take over their role. A cardiovascular system that has been running on a different configuration for 9 months rewires itself in minutes. And the first cry. Lungs that have never produced sound produce sound. A voice that has never been heard is heard for the first time, in the only language available to a human being in the first seconds of life, the language of arrival, of presence, of "I am here and I am alive." Minutes later, the second cry, different from the first. The same genetic blueprint, the same extraordinary journey from that single fertilized cell 9 months ago, the same beginning in every biological and molecular sense, and yet, indistinguishably, someone else entirely. 2 voices, 2 separate and complete human beings, 2 lives beginning simultaneously in the same moment and the same room, each one carrying within every cell of their body the memory of a beginning they shared.